Thank you very much once again for the invitation and for being here until the very last day. So we're going to switch the topic to a different subject, which is cardiac radiofrequency ablation. I don't know if you know about that, but I will try to explain what's going on here. So the object is to introduce a generalized theoretical and computational framework in order to account for thermo-electromechanical couplings at different scales during this clinical procedure. Basically, when we suffer of cardiac arrhythmias, and in particular cardiac fibrillation, it is necessary to act with different kinds of therapies. One of these therapies is cardiac ablation. And the object is to enter the cardiac chambers and to burn some specific portions of the tissue, such to avoid these electrical instabilities that cause arrhythmias and eventually deadly fibrillation. Of course, this procedure is very critical and requires a high control of what's going on into the tissue, such that we don't mess up everything. In these terms, of course, cardiac modeling is very much advanced. We know that is a multi-layered and multi-structured material characterized by a complex reinforcement of fibers, and these fibers act at different length scales. Here there is a representation of myocardial fibers that rotate through the ventricular wall, but we have also collagen fibers in between muscular fibers and so on. So this picture is only to tell you that it's very critical, the anisotropic contribution for the thermomechanical problem. And now, what is strange but missing in this kind of literature is exactly the role of anisotropy into the thermomechanical coupling and how this coupling is thermodynamically formulated. Okay? So the aim is to introduce to this kind of modeling. When we talk about radiofrequency ablation, we are talking about a specific phenomenon. We have a catheter. This catheter releases a certain amount of radiofrequency source. This radiofrequency source moves water inside the tissue, and at the same time, the catheter is pushed into the tissue to have the best contact possible between the catheter and the surface of the tissue, and this is for energy transmission. Then, of course, this generates heat, and heat kills the cells. So these are all the different physical phenomena that we should take into account. In order to start, we of course start simple. So we take idealized geometry, which has representative dimensions of a ventricle, so 8 centimeters by 8 centimeters by 2 centimeters. This is a representative geometry. And this is the aspect ratio with respect to the catheter. So boundary condition basically as very far away. Okay? Inside, we consider classical reinforcement of the myocardial fibers. So this is called rotational anisotropy. So this is basically a transverse isotropic material, and in each plane along the thickness we have a different reinforcement of the fibers with a different angle. Now, if I go on, okay, everything is framed in the classical continuum mechanics formulation. So you have the classical definition in finite elasticity. We assume the classical strain invariance of the deformation and the fourth pseudo invariant for the directional stretch along the muscular fibers. And these are the classical textbook definitions. What we have to consider is thermomechanics. Okay? In terms of kinematics, of course, we should also track the deformation induced by the additional role of temperature. And this is done in a very simple way, accounting for the multiplicative decomposition of the deformation gradient and using a very simple form of the thermal deformation map. Okay, it's the simplest that you can imagine. Even though, of course, whatever the tissue may be incompressible, of course, we do not impose incompressibility into the thermal map. So eventually we're going to have residual strains as what is expected from the experiments. Okay, from the mechanical point of view, we just impose equilibrium, so classical boundary conditions. Here we impose the traction forces from the electrode. The tissue is constrained at the bottom, but as I told you, mechanically, boundary conditions are very far away, so they don't play a critical role from the mechanical point of view. But what is critical is the material model. In fact, we have to consider all the different energetic contributions. In these energetic contributions, we have to account for the passive material behavior and its incompressibility, the hyperelasticity, due to the coupling with the temperature. So here, in this Mooney-Riving-like material, the stiffness-like parameters, they change with temperature, because as you cook a steak, the stiffness of the steak is going to change, and this effect must be considered. In the anisotropic part, once the tissue gets damaged because of a high temperature, fibers degrade, so the anisotropic contribution must decrease because of a damage of the tissue, and this is this term over here. And finally, this is the strain energy density for the thermal problem. Okay? In this particular energy, we also account for the cell death. And this is this C parameter that I'm going to describe soon after. In terms of the electrical model, we can assume that the tissue is purely resistive because radio frequency ablation works at 500 kilohertz, so the wavelength is way larger than the tissue size, so this relation holds, and we can just consider the electric contribution, not the electromagnetic contribution. So we can solve the quasi-static Maxwell equation, because magnetic contributions are not important. What is really critical here is this constraint. The total dissipated power within the tissue, which is the power delivered by the machine to the heart, must be fixed. Otherwise, we burn everything. Okay? This is a very critical constraint. If you don't respect this constraint, you are simulating something else. You are not simulating cardiac radiofrequency ablation. And then we have the classical boundary conditions. Here we tested different kinds of boundary conditions, but basically these specific Neumann boundary conditions are okay. Then we have to consider the transport of heat within the tissue, and we have to generalize the heat equation accounting for the deformation of the tissue. So here there is the so-called geometric coupling because of the flux must pass through a deformed domain, so the thermal conductivity tensor is pulled back into the reference configuration such to account for the deformability of the medium. And here as well, boundary conditions are very critical instead, because on this surface there is the wetting of the blood, and on the catheter surface there is the wetting of a saline solution coming out from the electrode. So here we have convective terms, and the other part we can assume Dirichlet constant temperature on the other side. Also in this case, everything is anisotropic, of course. Now, I pointed out here an arrow because we generalize also the heat capacity. Because we are dealing with a full thermomechanical problem, and in this case, the correct definition of heat capacity comes from the second derivative of the energy with respect to temperature. If you do that, you have a classical heat capacity at constant deformation, and this is what everyone uses usually, but you can also have an additional term because of the thermomechanical strain energy function, okay? And in this definition, again, the cell behavior, in particular cell death, is going to enter, okay? If the energy depends on cell dynamics, also this thermomechanical heat capacity is going to depend on the same effect. And in fact, this is the model that we use. This is a three-state model for the cellular death. Basically, we consider that cells can be in the alive state, so they are happy. But once we stress the cells with a higher temperature, they eventually pass through a vulnerable state. This vulnerable state in which proteins starts to unfold, they eventually can come back in the alive state if we remove temperature. If temperature is not there for long enough, they come back to the original state. Otherwise, they finally die, okay? So this is a very simple three-state model. And what's entering into the strain energy function is the relative shrinkage of the protein, which comes as a parameter coming from the different concentration of the three states. Okay? Of course, this is one of the possibilities. We can go with higher order multi-scale models. Okay. Okay. Then, as I was mentioning you, in the strain energy density, we also have the stiffness-like parameters that have to change with temperature because tissue gets stiffer as temperature increases, but also thermal conductivity and electrical conductivity, they may change with temperature. There are some evidences that this is the case, as well as the perfusion of the tissue. Whenever we burn the tissue, blood vessels, they close up, so you don't have any more perfusion. And the simplest thing is to say, once you have we reached a certain temperature, you just drop the perfusion coefficient to 0. This is one possibility, of course.
But you only can have the capillaries, because if you hit larger ones, it's a global, more global, no?
Yeah, this is exactly for capillaries. They close up when you burn the tissue.
Then it's clear, only capillary, if you hit another one.
Oh, no, of course, this is not for large vessels, otherwise we kill the patient. Yeah, no, these are for the small perfusion terms. Yes, absolutely. So basically everything is coupled with everything else. It's a multi-feed and multi-scale model that requires some complex computational framework. Everything was implemented in finite elements. We tested the convergence rate and so on. I don't want to enter the details. I want to just go to some of the results. So this is the clinical procedure. So you push the electrode and you have an initial stiffening because of the material response. Then you start the clinical procedure of radiofrequency ablation. As you start radiofrequency ablation, heat is going to accumulate into the depth of the tissue, but because of material properties are changing, the electrode is pushed back, okay? The stiffness of the material is increasing, so this is the tip of the electrode, reach a certain maximum, then by time, the tissue pushes back the electrode. So this is really critical because medical doctors want to get the highest possible surface to maximize the energy that they deliver. Otherwise, they are wasting time, and time is patient life. So this is another critical parameter that we have to take into account. Okay? And we tested for different weights, and this is always the case. But what's happening here is that basically we get a residual strain state, which is quite complex. And this is complex because of the anisotropy that we introduce in the electrical model and in the thermal model, other than the mechanical one. Okay? This is the shape, and the shape now can be compared with classical measurements adopted in clinics to say it was effective or not. Usually they measure the 50 degrees isothermal contour, saying that if I reach 50 degrees, I kill the tissue and I'm happy. Actually, this is not the case. In fact, within the 50 degrees isothermal contour, you both have a shrinked region, which is this one, and an expanded region, which is the one below. Basically, they come inside over here. See, this is shrink and this is expansion. Okay, so it's much more complex than just a 50-degree isothermal contour. Plus, we have residual strain over here. And we try to characterize this model in terms of versus experiments. Was not really a validation, was more let's see if we are going in the right direction. So we performed exactly the experiment. We measure in depth of the tissue with optical fibers the thermal state. These are 7 measurement points for 4 different sensors, so we have a lot of traces. And over all the traces in depth, what we observe is that anisotropy is really critical. This is just a classical Fourier model, and we are not matching the experiments. So we said, what's going on here? Probably we need something more, and probably we need a cation formulation with a generalized heat flux. And this, in fact, was the case. In the literature nowadays, it's called maybe a dual phase lag model, but coming from a Cattaneo equation. Basically, we introduced 2 additional terms. One, that they are related to 2 different time constants, right? And if you have 2 additional time constants, you can get nicely the experimental traces in average. Of course, this is the dispersion that you have from the experiments. But if you do not use a Cattaneo-like formulation, you don't get the 2 time constants, because here it's critical to have an initial rise and then a saturation effect. Okay? With a classical Fourier model, you don't get that. You only get eventually the saturation. Okay? So, and finally, we were interested in to understand what happens in terms of the anisotropy in space. These are experiments, okay? The measurement that we got in depth, and this is the model. And what is critical is really the fact that we need to introduce anisotropy in order to reproduce what's going on in the tissue. Otherwise, our estimates are totally failing. Okay, and that's it. We are going towards more complex contact models. We are trying to build up also for laser, similar laser applications, very tight control of temperature by monitoring the position of the sensors and so on. In this case, the formulation is even more complex because when you have a laser, there is interaction between scattering and optical features of the material. But more or less, this could be the arena in which we are working. And that's it. I mean, I hope I was fast enough for this time.